friends, today we are going to study about protein function in that protein interactions modulated by chemical energy. So muscle proteins are the proteins that are modulated by chemical energy. So what are the major muscle proteins? These are myosin and actin. And these proteins are arranged in filaments which slide past each other to bring about contraction. Now, structurally, the myosin looks like, as you can see in the diagram. This coiled portion, including this globular head, this is myosin. It has two terminal, the carboxyl terminal and the head is the amino terminal. Now, carboxyl terminal, if you see the structure, it reminds us alpha keratin. We also studied in the previous video of alpha keratin that it looks like a coiled coil structure. So here also the myosin looks like a coiled coil alpha keratin structure, the carboxyl terminal. Coming to its amino terminal, which has a globular head, this is the site where ATP is attached and hydrolyzed. The muscle contraction is a ATP dependent process and the myosin globular head is the site where ATP is hydrolyzed. Now, where is the light chain? These globular heads are attached with the light chain here. So, these light chains are associated with this globular head. Now, one more thing is this myosin, it consists of 6 subunits, among which 2 are heavy and 4 are light. When we treat myosin briefly with trypsin, it divides the myosin into two parts. It will break from here and divide the myosin into two parts. Now this part, which has its carboxyl terminal, it is known as light meromyosin. And the part including its globular head as well as the light chain, this is known as heavy meromyosin. Now this heavy meromyosin, when again treated with papain, it breaks the heavy meromyosin into two halves. One is the S2. S2 is the remaining coiled coil part and S1. These two S1s are the globular head to which light chains are attached. Now this head is called the motor domain. Why? Because this part only makes the muscle contraction possible, because this part contains ATP hydrolysis site. And as because muscle contraction is ATP dependent, so therefore this part is known as motor domain. In muscle, the molecules of myosin aggregate to form thick filament. This blue one is myosin. So as you can see, what is the structure of myosin? This coiled coil structure will have a globular head. So as you can see, multiple such coiled coil structures, each having a pair of globular heads, are arranged. And this full arrangement form a thick filament. So thick filament is mainly made up of myosin protein and in which multiple myosins are arranged in a regular fashion. Now this thick filament, it serves as a core of contractile unit. Now to form such type of thick filament, hundreds of myosin molecules are arranged and they form a long bipolar structure. The structure, as you can see in blue, this is known as long bipolar structure. And what else we can see? These globular heads are projecting outside. Everywhere you can see, these are the globular heads that are projecting in either direction in a regular manner. Now come to the actin. This orange one is the actin protein. These are abundant in almost all eukaryotic cells. Now here you can see this single circle, one single circle. These are known as G subunit. Now G subunits associate to form this complete polymer which is known as F-actin. The single molecules or the single monomeric actin molecule is known as G-actin subunit, and when multiple G-actin subunits are associated, that association is known as F-actin subunit. Now in this F-actin subunit, along with actin, two other proteins are present, troponin and tropomyosin. Now each actin monomer in this thin filament, it binds tightly and specifically to one myosin head group. So the main proteins are myosin and the actin. Myosin are arranged having two parts, the coiled coil filamentous part and the globular head, which contain the myosin head group along with the light chain. Now that globular head is the site where ATP is hydrolyzed, which brings the contraction possible. Now there are some additional proteins also that are organized in this thick and thin filaments. So let's first look how our muscle is arranged. So as you can see in the above figure, this is muscle. These bundles are the bundles of muscle fibers. That means our muscle is made up of bundles of muscle fiber. Now this is a single unit in that bundle of muscle fibers. What that unit contain? This is single muscle fiber which is made up of myofibrils. Now myofibrils has sarcoplasmic reticulum. Now this extended portion is a single myofibril. Now what this single myofibril contains? I band and A band, that is thick and the thin filament. And this combination of thick and thin filament is known as sarcomere. Now this I and A band, as you can see here, I band is the light band, that is thin filament. A band is it contains thick filament also, plus the portion where thick and thin filament overlap. Now in the middle of this I band is the Z disc. That means Z disc is the segment where thin filaments are anchored. Next is this M line in the center of A band. So it is a region of high electron density in the middle of the thick filament. I band is thin filament. Thin filament means mainly made up of actin. A band is thick filament. Plus it also contains where thick and thin filament overlap, mainly made up of myosin. Z disc where thin filament are anchored. M line, it is found in the middle of thin filament. This complete contractile unit is known as sarcomere. As you can see in this above diagram, the A band, I band, everything, this complete set is known as sarcomere. Now the sliding of this thick and thin filament cause shortening of sarcomere, due to which the muscle contracts. Now this Z disc where these thin filaments are anchored, it also contains some other minor proteins. What are these? Alpha-actinin, desmin, vimentin. This thin filament which contains mainly actin, along with actin, it contains a large protein called nibulin. M line, which is in the middle of thick filament, that means it should have myosin more, but along with myosin, it also contains paramyosin, C protein and M protein. These are in the M line. It is in the middle of thick filament, that means mainly it contains myosin, but along with myosin, it also contains paramyosin, C protein and M protein. Now one more protein is there which is called titin. It is the largest single polypeptide protein. What is its function? It links thick filament to Z disc. What titin do? It links thick filament to Z disc. Now this titin and this nibulin, these two proteins serve as a regulator, will regulate the length of the thin filament, and titin will regulate the length of thick filament. Now let's study how the contraction takes place. So as we now know that the contraction is a ATP dependent process. So in the absence of ATP, this first figure is the absence of ATP. So in the absence of ATP, what we can see is the myosin is tightly bound with the actin. So let's label this dark gray color actin molecule as A. Next, this light gray color is B. The next is C. So the myosin is bound tightly with the actin, that is A-actin, in the absence of ATP. Now as soon as the ATP will come and bind to myosin head, now you can see a small gap generated between actin and myosin. That means after binding of ATP, the bond between actin and myosin becomes weak. Now this ATP will get hydrolyzed into ADP and PI. Now as soon as this ATP get hydrolyzed to ADP and PI, as you can see in the next figure, the right side top, there occur a conformational change in the myosin head. What is that conformational change? Earlier, the myosin head is in contact with this A actin. Now, the myosin head stepped ahead and now it is in contact with this B light gray color of actin. But still, the interaction is a weak interaction because we can see a gap between these two molecules. So, due to conformational change, now the myosin head moves a step forward and now it is in contact with the next actin molecule, that is B, as we labeled for this figure. But the interaction is still a weak interaction. Now the next step will be the release of PI. As soon as the PI get released, one more conformational change will occur in the myosin head. Now you can see in the next diagram, the actin and myosin, now they are tightly bound. So now as soon as the PI get released, now this myosin head is tightly bound to the next actin molecule. After that, what will happen? The ADP will also release. As soon as the ADP will release, it will trigger a power stroke. Now the myosin head returns to its original resting state. So this how the contraction happens. The myosin will slide past the actin molecule. Now this step will again then get repeated. And that's how the contraction proceeds. That means sliding of thick filament along thin filament. Now this contraction should be highly regulated. Why so? It should not be in a way that any time that contraction is happening. It should be highly regulated. The contraction should only happen when there is a signal, when there is a nerve impulse. So this is controlled by two proteins known as tropomyosin and troponin. In the last slide, we already studied that this tropomyosin and troponin, these are present in thin filament along with actin. Now this troponin is a calcium binding protein. So in the absence of any signal, this tropomyosin blocks the myosin binding with actin. As soon as nerve impulse is generated, what it will do? Because of this signal, the calcium will release from sarcoplasmic reticulum. This released calcium will then go and bind to troponin, and this binding will cause a conformational change in this troponin-tropomyosin complex. As soon as this conformational will change, this tropomyosin will then no longer able to block the myosin. So now the myosin binding site is free, so it can bind with the actin and the contraction will start. So the process is highly regulated. It won't happen until there is a signal. So once the signal is released, because of that signal, the calcium will release from sarcoplasmic reticulum. This calcium will go and bind to the troponin. Because of this binding, the troponin-tropomyosin complex conformational change will occur. And because of this conformational change, the tropomyosin will leave the myosin binding site. So now the myosin binding site is free to bind with the actin to proceed the contraction process. So this is all about how muscle contraction occurs. Now one question from my end for you all, that is, when a higher vertebrate dies, its muscles stiffen as they are deprived of ATP, a state called rigor mortis. Explain the molecular basis of rigor mortis. Do let me know in the comment section. If you found the video informative, please do like, share and subscribe the channel and hit the bell icon for the next video notification. Till then, stay safe, stay happy.